Hello, and welcome to the third installment of our lectures on the cardiovascular system for Columbia College Anatomy and Physiology, Biology 130. This lecture, the first lecture on the cardiovascular system, we discussed the heart. The second lecture on the cardiovascular system, we discussed the blood vessels. And the third lecture on the cardiovascular system, we're going to discuss the blood itself. You can find this material in chapter 18 of the book, but probably, as I've said, that chapter is a little bit more detailed than you would need. So just concentrate on the lecture slides, which are still available on your website. All right, so an overview of what I'm going to talk about. I'm going to start by talking about the process of hematopoiesis. The process of hematopoiesis means the formation of blood cells, both red and white blood cells, from bone marrow cells, the little gooey stuff that you find inside flat bones, is where hematopoiesis takes place. So that means these cells that you find in flat bones are referred to as hematopoietic stem cells. And through various combinations of cytokines and growth factors, which are little small proteins that operate as intercellular signals, if a hematopoietic stem cell is bombarded with the right combination of cytokines and growth factors, it will turn into an erythrocyte, a red blood cell, or a leukocyte, a white blood cell. And then there are several different types of leukocytes, several different types of white blood cells that it could differentiate into. The correct term is differentiation. So when you go from a kind of a generic cell to a specific specialized cell, that process is called differentiation. Differentiation is usually terminal and in one direction and terminal. So a cell, if you have a general bone marrow cell, a hematopoietic stem cell, and it differentiates into a granulocyte of some sort, that is considered to be terminal differentiation. It can never go back to being a bone marrow cell again. All right, then after we've discussed hematopoiesis a little bit, we're going to discuss the life cycle of an erythrocyte. An erythrocyte, of course, is a red blood cell. The fact that they are made in the bone marrow and then they circulate around the bloodstream for a few months and then they are destroyed in the spleen. We will discuss the two different lineages of the blood cells. So you probably know that a lineage is a fancy term for your ancestors or your family. So you say, what is your lineage? That refers to who is your family? Who are your ancestors? Where do they come from? And all of the blood cells belong to one of two lineages. The myeloid lineage contains the red blood cells as well as some of the leukocytes. And the lymphoid lineage contains the B cells and the T cells, lymphatic cells, and a few others which we're actually not going to talk about in this part of the course. But for now, B cells and T cells are critical parts of the immune system, and they are one type of leukocyte, and they belong to the lymphoid lineage, as opposed to the myeloid lineage, which contains granulocytes, erythrocytes, platelets, or thrombocytes, and a few other things. Then we'll take a little side detour to explain to you what happens to the bone marrow, which is responsible for creating the blood. What happens to the bone marrow, the hematopoietic stem cells, during cancer treatment? Cancer treatment actually, different types of cancer treatment actually kill the bone marrow cells, and you may have to have them replaced through a transplant. Okay, then we'll talk about what different types of blood cells can you expect to find in the blood. Some of them are white blood cells, the leukocytes, and some of them are red blood cells, the erythrocytes, but there are several types. And of course, we will have already discussed what they are in the context of what lineage do they belong to. Then we'll talk about something that's very important to emergency medicine, for instance, and other things, which is called the hematocrit. The hematocrit specifically is if you take the volume of blood and then you take the percentage of that volume that is taken up by the red blood cells and the white blood cells as well. So you take the volume of blood and then you take the percentage of that volume that's made up by the cells. That is referred to as the hematocrit. And the hematocrit is typically something like 45%. So that means that around 45% of our blood is made up of the cells, and then the rest of it, the other 65%, is the serum, the liquid part. All right, and then we're going to talk about something called hemostasis. Don't confuse the word hemostasis with the word hematopoiesis because they mean two slightly different things. The word hemostasis refers to blood clotting. Right, so we're going to talk about platelets, or the technical term for a platelet is called a thrombocyte. And we'll talk about how thrombocytes will plug holes in the blood vessels by forming a clot. And then we will talk about the ABO blood grouping system, which again is important to medicine and things like that, blood transplants. You probably know that you can't transplant type A blood into a type O recipient because the type O recipient's immune system will reject and destroy those red blood cells. In fact, you can only transplant type O blood into a type O recipient, and we'll discuss why that works. And then we'll talk about some various pathologies of the blood cells. We'll talk about anemia. The term anemia specifically refers to a lack of red blood cells only. And then we'll talk about other things like hemophilia. Hemophilia is a malfunctioning of the thrombocytes and the factors that function from the thrombocytes, the blood clotting factors, so that hemophilia is characterized by a problem with your blood clotting. If you cut yourself, your blood takes a rather long time to clot if you have hemophilia. All right, so hematopoiesis, also sometimes it is sometimes called hemopoiesis, which is less common, but the more technically correct name is hematopoiesis. Don't confuse the term hematopoiesis with hemostasis. As I said, hematopoiesis refers to the formation of blood cells. Hemostasis refers to the process of blood clotting. All right, so blood cells are created, hematopoiesis, in the flat bones. In an adult, that means the bone marrow that is found inside the clavicle, inside the skull, the spine, the ribs, the sternum, and the pelvis. And the heads of the femurs of the humerus, the humerus bone also contains a little bit of spongy red bone marrow. It's referred to as red bone marrow because if you ever break open a rib, you'll see that it's red inside, and that's because some of those bone marrow cells that are inside the bone are converting into erythrocytes, and so they are red in color. Now, all of these blood cells originally come from a type of a progenitor cell, a type of original ancestral cell called a hematopoietic stem cell. A hematopoietic stem cell is sometimes commonly referred to as a bone marrow stem cell, right? So all of the red and white blood cells originally come from a hematopoietic stem cell or a bone marrow stem cell. And they are signaled from other cells. They receive signals in the form of cytokines and growth factors, and in some cases hormones. They're signaled from other cells to differentiate into various types of blood cells. Differentiation refers to turning into, to become or to turn into another type of cell. Right. And these cytokines and growth factors that tell these hematopoietic stem cells to differentiate into erythrocytes or leukocytes, they are mostly peptides, meaning short proteins, little tiny short proteins that operate as intercellular signals. A signal sent from one cell to another. They don't send signals to each other with radio waves or television signals. They send signals to each other by sending out little proteins that will interact with protein receptors on the surface of the target cell. So these are the bones bone marrow where the blood cells are formed, the flat bones. And at the top of this is a flow chart which shows all the different types of bones that all the different types of blood cells that you can have down here at the bottom. Right, so we have platelets down here. We have eosinophils, granulocytes, red blood cells or erythrocytes. And over here we have T cells and B cells. Well, all of these cells originated from hematopoietic stem cells. And these hematopoietic stem cells were stimulated by some of them were stimulated by one set of cytokines that caused them to enter what's known as the myeloid lineage. And then once they've entered the myeloid family or lineage, if you stimulate them with some other cytokines like stem cell factor and thrombopoietin, which is what those letters stand for, they will differentiate into thrombocytes or platelets, for instance. So the different cytokines that are used to turn different cells into turn the general cell into a specialist cell are indicated with these letters here. Another example would be you have a hematopoietic stem cell, and then you stimulate it to enter the myeloid lineage, and then you stimulate it with a cytokine called erythropoietin, and that causes that general cell to differentiate into an erythrocyte, a red blood cell. And the same thing is true for all the other types of blood cells, except that we stimulate them with different cytokines, and that tells them what they're supposed to turn into. Right? It's kind of like a student. You have a student on the first day of college and they haven't studied anything, but if you get them to study these courses, they will get a degree in this subject, as opposed to these courses, which give them a degree in this subject. Stem cells, bone marrow stem cells, hematopoietic stem cells, are said to be pluripotent, which means that they have the ability to turn into any of the blood cells, but not into any of the cells in the body in general. The difference between pluripotent and totipotent, which you see down here, totipotent cells have the ability to turn into any type of cell in the body. Right? So hematopoietic stem cells can turn into any one of the blood cells. That's called a pluripotent stem cell, or pluripotency. Totipotent cells have the ability to turn into any cell in the entire body, and there's only one place that you would find these cells, and that is embryos, human embryos or animal embryos or animal blastulas. So if you take a cell out of a blastula, those cells are general or generic, and you could use them to they could actually have the ability to turn into any type of cell in the mature body. Right, so there's a lot of research going on about totipotent embryonic stem cells and what kinds of things you can do with them and so on. All right, let's look at a specific example called erythropoiesis. Erythropoiesis is a subcategory of hematopoiesis. Erythropoiesis refers specifically to the formation of red blood cells. Okay, there are special cells in the liver and the kidneys that are able to sense how much oxygen is in the blood when the blood flows through the liver and the kidneys. Right? Now, if these special cells in the liver and the kidneys sense that the concentration of oxygen is too low in the blood, they assume it's because there are too few red blood cells. Right? They assume it's because there aren't enough red blood cells. So these specialized cells produce a cytokine called erythropoietin. Erythropoietin is abbreviated EPO, erythropoietin. Erythropoietin travels through the blood and eventually makes its way to the red bone marrow and then signals the hematopoietic stem cells to differentiate into red blood cells. Initially, these blood cells differentiate into a cell type that's called a reticulocyte, and a reticulocyte is actually a live cell. Now, the reticulocyte will turn on the genes for hemoglobin, and the reticulocyte will fill up with hemoglobin. Hemoglobin, of course, is the protein that's used to carry oxygen around the body. And then once the cells are filled to the brim with hemoglobin, they die, and the nucleus disappears. So the nucleus disappears because these cells are essentially dead bags full of hemoglobin. And because the nucleus dissolved, they kind of flatten out. And so they have this kind of a flattened donut shape. And those are the mature red blood cells. So I could ask you on the final exam, what is a reticulocyte? Or I could ask you, what is the name of an immature red blood cell before the nucleus dies, before the cell actually dies and the nucleus disappears? All right, so that is how red blood cells are created. The liver cells in the liver and kidneys are able to sense that there's not enough oxygen in the blood. That may be a mistake. These cells assume that the reason there's not enough oxygen in the blood is because you don't have enough red blood cells, and then they send a signal to the bone marrow for more of these cells to turn into red blood cells, and that's how you get more. So as I said, in a mature red blood cell, the nucleus dies and dissolves, and so the center of the cell kind of collapses like this and flattens out. So that's why you have these kind of flattened shapes, because the nucleus is dissolved and they kind of deflate like that. This is a microscope image. These are erythrocytes. These are white blood cells, or leukocytes, and we're going to talk about them next. But this is what these cells here are what red blood cells look like. and I'd like to direct your attention to the central part of the red blood cell. You notice how the center of this red blood cell looks lighter than the rest? The reason for that is because these red blood cells are sitting on a microscope slide with light passing upwards through the light source, passing up through the slide into your eye. And at the very center of the red blood cell, they are thinner because the nucleus disappeared, and so they're kind of collapsed in that area. So that corresponds to this central area here. And we call that a zone of central pallor. Zone means an area, pallor means pale, and central means in the middle. So there's a middle part that is paler or lighter than the rest of the red blood cell. We call that a zone of central pallor. That term will become important in a minute when we talk about blood diseases, because you use the size of that zone of central pallor to tell whether the red blood cell is full of hemoglobin or whether it's empty, or some other diseases, some other problems. Okay, so red blood cells are kind of flattened out. They have a zone of central pallor, which is because the nucleus is dissolved, and so the center of the cell has kind of collapsed. Here's hemoglobin. This is the structure of hemoglobin. So hemoglobin is made of, basically it's made of a globulin protein with a prosthetic group called heme. So the prosthetic group, which is called heme, is a complicated chemical, but it's not a protein. It's not made of amino acids. That's what makes it a prosthetic group. So we talked about prosthetic groups earlier in the course. And notice that at the very center of the heme group is an iron atom, an iron ion actually. And that iron ion is able to carry oxygen around. So at a certain pH, at a high pH of 7.4, oxygen will bind to that iron ion, and then the blood will travel down to the muscles and the other organs of the body where the pH goes down. And when it reaches an area where a muscle is working hard, the muscle is producing a lot of carbon dioxide, which goes into the blood. Extra carbon dioxide in the blood causes the blood pH to go down to 7.2 or even lower than that. The iron ion will let go of the oxygen. Right? So the oxygen will bind reversibly to iron in the heme prosthetic group depending on the pH. All right, so here's a question for you about, now that you know how red blood cells are formed by the body, the kidneys, and the liver producing erythropoietin, you may know that some athletes cheat, especially endurance athletes like bicyclists and marathon runners. They occasionally cheat by injecting themselves with artificial erythropoietin. And that's a favorite way for them to cheat, but can you imagine how it would be? Right? Why would that give you a competitive advantage? Right? So you can stop the video and just think about that for a minute and then see if you have the answer right. But the answer, for example, Lance Armstrong was a famous bicycle racer doing the Tour de France, and he won the Tour de France 7 times. And here he is demonstrating how many times he cheated to win the medals. Right? He injected himself with erythropoietin. And I'm totally surprised nobody figured that out, because here you had a man who was in his 40s who was beating men that were in their 20s in bicycle races. That doesn't sound natural. But in fact, they figured out eventually that he was injecting himself with erythropoietin. That has the effect of increasing the number of red blood cells in your blood, which means that your blood can carry more oxygen than it otherwise could. So if your blood can carry more oxygen around to the muscles, then your muscles can work harder because they're getting more oxygen. So injecting erythropoietin is a standard way for certain athletes to cheat. All right, so what are the dangers associated with injecting erythropoietin? Are there some dangers, do you think? Well, in fact, there is a danger, and I'm gonna ask you to think about. I explained earlier what hematocrit is. Hematocrit is the percentage of your blood that is taken up by red blood cells. And if you increase the hematocrit, if you inject yourself with erythropoietin, what would be the effect on the hematocrit? It would increase, wouldn't it? So your hematocrit might go from 45% to 50 or 55 or 60%, and then pretty soon it becomes very difficult for your heart to pump this blood around because it's so viscous, it's so thick. So somebody once made the analogy. They said if you raise the hematocrit up to about 65% because your blood contains more red blood cells, the heart has to pump this liquid through the veins and the arteries. That's kind of like pumping cottage cheese through a garden hose. I think that's a good analogy. You can imagine that a pump that's pumping water, suddenly it has to pump cottage cheese through a garden hose instead of water. It's going to be working a lot harder. So every now and then, tragically, athletes that have been cheating by blood doping, they drop dead from a heart attack because their heart is in, drop dead from a heart attack in your 20s because your heart is having to work too hard pumping that extra thick blood around. All right, so let's look at the life cycle of an erythrocyte once it's created. Okay, so erythrocytes are created in the red bone marrow in response to erythropoietin. The stem cells differentiate into a reticulocyte. They fill up with hemoglobin, they die, and then they flatten out. They have no nucleus. They circulate around the bloodstream for about 120 days. And then the bloodstream travels through an organ called the spleen, and the spleen is able to, the spleen is kind of a filter that filters out bad stuff from the blood. And the spleen is able to sense whether the red blood cells are smooth or rough on the surface. If you have young, healthy, newly created red blood cells, they have a very smooth surface. If they have a rough surface, it suggests that they're worn out. And when you have worn out red blood cells passing through the spleen will destroy them and break them down and recycle the components. So the hemoglobin inside those blood cells is broken down into amino acids and then put back into the blood. The iron is transported to the liver where it's used for other things. The heme group is sent to the heme group is transported to the liver as well. And so the spleen breaks down the components and uses them for other purposes. Now, when we talk about some of the anemias, some of the blood pathologies later, we'll discuss the fact that sometimes the spleen breaks down those blood cells mistakenly because they have a funny shape, and the spleen mistakenly thinks they're worn out when in fact they're not worn out, they're brand new. Sickle cell anemia is a good example of that. That's where you have there's a mistake in the hemoglobin molecule that causes the blood cells to assume a sickle shape. If you've ever seen a sickle shape or a crescent moon shape, you know that it has two sharp edges on either side. The spleen senses these sharp edges on the blood cells and it destroys them, even though they're not worn out. They're perfectly fine. They're just a funny shape. So that's one form of anemia, and there are others where the spleen mistakenly destroys perfectly good red blood cells. All right, so hemoglobin, hemoglobin is made of 4 globin proteins, which are encoded by 2 different genes, and a heme group with an iron in the center. Right, so we've seen this already. Globin is a protein, so in the spleen, the globin is broken down into amino acids. The iron and the heme group are carried to the liver by a protein called transferrin, right, which is also a globular protein. And from there, the iron is stored in the liver, but the heme group is broken down into a chemical called biliverdin, and then later to a chemical called bilirubin, which is kind of a yellow-brown color. And that biliverdin is carried to the liver by albumin, and then it gets put into the bile, which is part of the digestive system, which we'll talk about later, and it gives feces that kind of yellow-brown color that it has, right? So when you eat food, it's all different colors. When it comes out, it's kind of brown color, and the reason for that is because it's been mixed with bilirubin. All right, now let's talk specifically about the different families, the two special families or ancestral groups that blood cells come from, and they are assigned to, the blood cell lineages. Of course, a lineage is your family or your ancestors. Blood cells have 2 families. They have 2 sets of ancestors. They have 2 lineages. One set is called the myeloid lineage, which contains the erythrocytes, the red blood cells, and some granulocytes, which are one of the forms of white blood cells, and also the platelets, also known as the thrombocytes, which are involved in blood clotting. The other lineage is the lymphoid lineage, which contains the B cells and the T cells, which are red blood cells that are part of the immune system. And it also produces a few other types of cells, which we're not going to go into in this section of the course. We might go into it in a later section of the course. All right, now, white blood cells, all white blood cells are called leukocytes. All white blood cells are called leukocytes because the word leuko means white. But there are two types of leukocytes. The lymphocytes, which are from the lymphoid lineage, and then the granulocytes are the white blood cells that come from the myeloid lineage. And they are called granulocytes because they look grainy when you see them under the microscope. They look grainy. Right, and we'll talk about what that means. Okay, so here we have a hematopoietic stem cell at the top of this flow chart. If it gets stimulated with certain cytokines, it ends up going into the lymphoid lineage and then becoming a B cell or a T cell. On the other hand, if it's stimulated with other cytokines, it enters the myeloid lineage and then it becomes either an erythrocyte or a platelet, a thrombocyte, or one of many types of things that are called granulocytes. These things here are called granulocytes. And if you look at these drawings, these cartoons, you'll see that if you look at the cytoplasm, you see these little spots in the cytoplasm. Right? These little spots or granules are vesicles that are filled with either chemicals or proteins that the particular granulocyte in question needs to do its job. And another thing to look at, another thing to notice about these cells, these granulocytes, is that the nuclei are really funny shapes. Funny shapes, right? So a nucleus is supposed to be round like you see here in the B cell or the T cell. But in a monocyte, for instance, it's kind of shaped like that. In a neutrophil, it's shaped like that. In an eosinophil, it's shaped like this. And whenever you have these different shaped nuclei, these cells, these granulocytes, are also referred to as PMN cells. PMN for polymorphonuclear. Poly means many, morpho means shape, and then nuclear means nucleus. So polymorphonuclear cells are granulocytes that have funny shaped nuclei. They have many different shapes for their nuclei. So those are the PMN cells. And the PMN cells, or the granulocytes, they're members of the myeloid lineage. Here's our chart again showing hematopoiesis, right? So depending on what cytokines you use to stimulate the hematopoietic stem cell, it'll enter either the myeloid lineage or the lymphoid lineage. And then beyond that, depending on what cytokines you stimulate them with, they will turn into any of these cells or any of these cells. Right, so on the exam, on the final exam, I could very well ask you what lineage do the erythrocytes belong to, or what lineage do the eosinophils belong to, what lineage do the thrombocytes belong to, and so on. Those are perfectly fair questions. All right, so some of the examples of the cytokines and growth factors that cause these things to differentiate into different types of blood cells. Erythropoietin, of course, we've already heard, that is produced by liver and kidney cells and causes the hematopoietic stem cells to differentiate into erythrocytes. Thrombopoietin, TPO, is one of the cytokines that stimulates a stem cell to differentiate into a larger cell called a megakaryocyte. Megakaryocyte is the precursor or the progenitor to the platelets. And so the platelets are actually not real cells, and they're quite small. So they're not real cells. They don't have a nucleus. They don't have a lot of things. The platelets are actually fragments of cells that broke off of a megakaryocyte. So platelets come from megakaryocytes, and their job is to cause the blood to clot. Okay, there's another cytokine called colony stimulating factor, which stimulates certain white blood cells to differentiate into, to various cells. A lot of the cytokines that tell cells to turn into this cell or that cell are referred to as the interleukins. And inter, of course, means in between cells, and then leukin means white. So interleukins literally means in between white blood cells. We have white blood cells that are sending protein signals to each other, telling them to differentiate into a slightly different type of cell or something like that. And the interleukins, we've discovered quite a lot of them, and we got bored of naming them, so we just give them numbers. So IL-1 is interleukin 1, IL-2 is interleukin 2, and IL-3, and so on. Right? And they are involved in the formation of various white blood cells. And then stem cell factor is responsible for stem cell self-renewal. Now what that means is if a white blood cell gets stimulated to differentiate into an erythrocyte, If that cell differentiates into an erythrocyte, then you've lost one of your, it goes away into the blood, and then you've lost one of your hematopoietic stem cells, haven't you? So before it does that, it basically divides in half, and then one of the two, whenever a cell divides in half, you call the two progeny cells daughter cells, not son cells, but daughter cells. One of the two daughter cells turns into an erythrocyte, and the other one stays as a hematopoietic stem cell again. So that way, we're not losing our bone marrow stem cells, because every time one of them is told to divide, it splits in half first with the help of stem cell factor, and then it's replaced, so it makes its own replacement before it leaves, basically, right? So stem cell factor, SCF. Just as a personal anecdote, I spent about 7 years of my life working on SCF, which I think now is kind of sad, but it was how I spent my 30s, was studying stem cell factor. I've moved on since then. All right, so that is how we do this. Okay, on a side note, let's talk about how cancer treatments affect hematopoietic stem cells. Okay, so generally there are two main cancer treatments. One is called chemotherapy. This is where you treat a patient with chemicals that target the cancer cells, and the other one is radiation therapy, where you treat a patient with radiation that targets the cancer cells. Okay, now if you have cells inside the body, let's say you have a bacterial infection, there are a lot of things, you have a bacterial infection that will make you sick. There are a lot of things that will kill the bacteria. You have to get rid of the bacteria, and there are a lot of things that will kill the bacteria. Unfortunately, most of those things will kill the human that the bacteria are inside as well. So the trick about curing somebody of a bacterial infection is that you have to find something, a drug or a chemical, that will kill only the bacteria cell without killing the human host, without killing the human that the bacteria is living inside. The same thing is true for cancer. How are you going to kill just the cancer cells without killing the person that the cancer cells are in? Right? Well, we haven't really made any new techniques or new advances on how to do that in the last 50 or 60 years or 70 years. We're still just kind of refining the two different techniques that we usually use to specifically target cancer cells, and that is by targeting the DNA as the cell is in the process of dividing. So in the human body, in the adult human body, very few of the cells are actually dividing. If you have chemicals that specifically shatter or destroy the DNA of cells as they're dividing, then you will have selectively killed just the cancer cells. And there are one or two other cells in the human body that are also dividing all the time. And unfortunately, bone marrow cells are one of those, right? So chemotherapy destroys DNA as it's being replicated in preparation for the cell to divide. Radiation therapy shatters, high energy radiation will shatter DNA. That doesn't matter to many of the cells if they're not dividing, but if you're attempting to divide, you have to make a copy of your DNA. And if you smash or shatter the DNA with high energy radiation as the cell is dividing, it stops and basically kills it. So in the adult human body, this is usually not a problem, because most of the cells are not dividing. They're just sitting there metabolizing. They're sitting there generating energy and producing proteins, but they're not dividing, right? Cancer cells are dividing, and so chemotherapy and radiation therapy will selectively kill cancer cells. But there are one or two examples of cells in the adult human body that actually are dividing, and chemotherapy and radiation therapy will kill them as well. And those things are the side effects of chemotherapy and radiation therapy. So what are most of the cells in the adult are not dividing. The ones that are dividing include the hair follicles, right? So one of the side effects of cancer is that your hair falls out. Another cell that's constantly dividing are the epithelial cells that line the gut in the intestine. So another side effect of chemotherapy and radiation therapy cancer treatment is that you have nausea, because your gut cells are being destroyed. Your skin cells are dividing constantly and being replaced, but if you suddenly stop that process for a while, it doesn't affect you too badly. And then the big one, of course, is the bone marrow cells, which give rise to your blood cells. So people who are undergoing chemotherapy or radiation therapy in order to kill cancer will be anemic, right? So the red blood cells you won't have as many red blood cells, because you destroyed a whole bunch of the red blood cells. You destroyed a whole bunch of the hematopoietic stem cells in the bone marrow. So then you have a lowered ability. You end up making much fewer red blood cells, and you end up making much fewer white blood cells. The white blood cells, of course, are involved in the immune system, so you're vulnerable to being infected by things that your immune system would normally destroy. Right? So the side effects of cancer include hair loss, nausea, skin cells, we don't care about that, and anemia, not having as many red blood cells, and also having a weakened immune system. So people who are undergoing cancer treatment have to be very careful that they stay in an environment that's not very dirty. They don't want to go out and be swimming in dirty, muddy water or anything like that, because you can get infected very easily if you're undergoing cancer treatment. All right, that's just a little side story. So chemotherapy and radiation therapy destroy dividing cells. You also kill the stem cells during cancer treatment. Now, what do you do if you destroy somebody's immune system in an effort to cure them of the cancer? Okay, well, that's what we call a Faustian bargain. Faustian bargain is something that you do now. It's from the Marlowe's wonderful play, one of my favorite plays, the Dr. Faustus. It's about a doctor who wants to cure people of the plague, so he makes a deal with the devil and says, "I'll give you give me magical powers that I can use to cure diseases and so on. You give me these magical powers for 20 years, and then you can have my soul." And then during the first 20 years, that seems like a good bargain, but then when the devil comes for your soul, it's not such a good bargain. So somebody's has cancer, you can kill the cancer with radiation therapy or immune therapy, but then in the process, you'll kill their immune system as well. And so what do we do about that? Well, one of the things that you can do is you can have a bone marrow stem cell transplant. You can have a bone marrow transplant, right? So there are various ways that you can take your own bone marrow cells and try and purify any cancer cells away from them, and then save them, and then basically irradiate somebody and kill their cancer and simultaneously kill their bone marrow cells, and then you put your own bone marrow stem cells back in. So when you give yourself a transplant, when you transplant yourself with your own tissue, that's referred to as an autograft. Sorry, right? So that's kind of rare, but it does happen. What's more common is that they will take bone marrow from a close relative, like a parent or a sibling, a brother and a sister, and then they put that into the person who's had their immune system destroyed, and that's called an allograft. So whenever you take a body part from somebody else and transplant it into yourself, that's called an allograft. Whenever you take your own body parts and move them from one part of the body to the other, that's called an autograft. Okay, if you're collecting bone marrow from your brother or your sister to inject into you, there are several places where you normally get it from. I could ask you this on the final exam. So some of the places that you aspirate, the word aspirate means to suck out. So there's some places where you would aspirate bone marrow from the donor. One is the sternum, right? So there's red bone marrow in there. Hematopoiesis is going on there. Taking it from the tibia, the head of the tibia is another typical place. Taking it from the posterior iliac crest of the pelvis is another place. So here's somebody who's having that done. They're sticking a needle into their hip and sucking bone marrow out to give that bone marrow to a cancer patient. Or the anterior iliac crest of the pelvis, right? So these are four places that you can aspirate bone marrow from and transfer it into a patient who's had cancer therapy. You can do so this is an example of an allograft, but you can also do an autograft. Right? One of the things that people are doing these days is that for certain types of cancer, when a baby is born, the umbilical cord contains their own stem cells, and that is referred to as cord blood. So the blood that's in the umbilical cord can actually be used to repopulate the immune system if you have it destroyed due to cancer therapy or radiation therapy. Problem is, nobody used to save that. Now people are starting to do that. So there are umbilical cord banks, biobanks they're called, where when your baby is born, you freeze down the baby's umbilical cord so that if they develop cancer when they're middle-aged, you can go back and get it. You can irradiate that person and kill the cancer, and then you can replace their bone marrow by going back and getting their frozen umbilical cord. Right, so that's one of the sources for stem cells is also the umbilical cord. So it's becoming fashionable nowadays, although it's very expensive, but it's becoming fashionable these days for people to freeze their baby's umbilical cord with one of these biobanks. Right? Autograft versus allograft. Okay, now let's talk about the different types of blood cells that make up the blood. Of course, we know from the myeloid lineage, we know about the erythrocytes. They carry oxygen. The thrombocytes are the platelets whose job it is to clot the blood. And then we have granulocytes. These are white blood cells that have a funny shaped nucleus. Therefore, they are the PMN cells. And they form part of the immune system. Right now, I'm just going to go over some of the major classes of PMN cells, the granulocytes. Monocytes are white blood cells that are involved in the immune system. Their job is to eat bad guys. Bacteria gets into your blood, it's eaten by a monocyte. Whenever a cell eats something in order to destroy it, that's referred to as phagocytosis. So cells that carry out phagocytosis are referred to as a class of cell called phagocytes. Notice that there's another bunch of cells here, the neutrophils. Neutrophils is another class of phagocyte cells. So there are several white blood cells that are classified as PMN cells, polymorphonuclear cells, that are also grainy in appearance, so they're granulocytes. And their function is to eat foreign invaders like bacteria and viruses that get into your system. Okay, then there's another type of a granulocyte called a basophil and another type called a mast cell. Those two types of granulocytes, those two types of leukocyte granulocytes are used in an immunology process called inflammation. And we'll talk about that when we get to the immune system. But basically, inflammation is when you have an infection or you have an injury of some sort and a certain part of your body becomes inflamed. And so, for instance, if the meninges, which we've already learned, the meninges are the membranes around the brain. If the meninges become inflamed, you simply take the suffix itis and add it to the end of the organ that's affected. So if you have infected, the organ that's inflamed rather. And so if you have inflamed meninges, we call that meningitis. If you have an inflamed appendix, you call that an appendicitis or an appendicitis. So that's that response, that inflammation is composed of heat, a feeling of heat, redness, swelling, and pain. Those are the four characteristics of an inflamed organ or an inflamed body part. Some of those responses, heat, pain, redness, and swelling, are mediated by the basophils and the mast cells. And we'll talk about that when we get to the immune system lecture. Finally, there's a granulocyte called an eosinophil. Okay, now eos, the word eos is an ancient Greek word for pink. So you know that leuko is the ancient Greek word for white. Erythro is the ancient Greek word for red. Eos is the ancient Greek word for pink. So these cells are grainy in appearance and they appear pink when you look at them under the microscope. And their job is to fight and destroy parasitic worms, microscopic nematodes, for instance, like Wuchereria bancrofti that causes elephantiasis. Whenever you get infected by a nematode or some other kind of a helminth, these are the microscopic worms that can infect you. The job of the eosinophils is to go out and destroy those worms before they can give you a deep infection, which usually works fairly well. All right, so these are all part of the granulocytes are all part of the immune system. The lymphoid lineage, T cells and B cells. B cells can differentiate into a type of cell called a plasma cell that produces antibodies. You've probably heard of antibodies. They are used to fight foreign infections, right? And so the B cells, which are capable of differentiating into the plasma cells, which produce the antibodies, are part of the lymphoid lineage. The T cells are another type of white blood cell. They coordinate the immune response. In fact, they spend a lot of their time telling the B cells what to do. And so those two types of cells are part of the immune system, but they are white blood cells. They are leukocytes. They're leukocytes, but they're not granulocytes. They're leukocytes, and they belong to the lymphoid lineage. Okay, again, the immune system. Okay, so you already know what these are. These are erythrocytes, and there's the zone of central pallor that takes up about 1/3 of the cell. Okay, what do you think these are? Notice that they're very small. Right? If you guess that those are platelets or thrombocytes, you are correct. Those are platelets, which are fragments of a megakaryocyte. Okay, now over here we have a granulocyte. It's easily distinguished as a granulocyte because it has a polymorphic nucleus. The nucleus is a funny shape. And over here we have a white blood cell that doesn't have a funny shaped nucleus. The nucleus is nice and round and it takes up most of the cytoplasm, and that is a lymphocyte. That's a B cell or a T cell. You can't tell which one it is just by looking at it, but this is a B cell or a T cell from the lymphoid lineage. This is obviously a granulocyte. You can tell from which is a member of the myeloid lineage. You can tell from the funny shaped PMN nucleus. And these, because they're small, must be thrombocytes. And then this stain is using a type of stain called Wright stain that stains erythrocytes kind of a purple color, and then it stains DNA a dark blue color like this. So you can easily tell lymphocytes from erythrocytes or leukocytes from erythrocytes because they stain different colors. So this is a stain, a microscope stain called Wright's stain. All right, so we have two types of white blood cells. One of them, the granulocytes, which belongs to the myeloid lineage, and the lymphoid cells, the B and T cells that belong to the lymphoid lineage. All right, so we've seen what they look like. All right, let's discuss hematocrit as promised. Hematocrit is literally the percentage of the volume of blood that is taken up by cells. Now, if you take a sample of blood, if you take a test tube full of blood, we already learned the fact from an earlier lecture that a test tube full of blood is called a vacutainer, which would be drawn out by a technician called a phlebotomist. And if you take that test tube full of blood and you leave it in a test tube holder sitting on the bench, after 8 or 10 or 12 hours, it will actually, the red blood cells and the white blood cells will actually settle to the bottom of the tube, and the serum will be sitting at the top of the tube. So they will separate given enough time simply by gravity. But you know that they make a machine called a centrifuge, and a centrifuge, you put these test tubes into a centrifuge and you spin them at a high speed, and it causes the serum and the cells to separate much faster in 5 minutes, right? So usually if you take a sample of human blood and you spin it in a centrifuge, you will actually see the difference between the serum at the top and the red blood cells at the bottom. And these things in the middle are the white blood cells. This particular, if you're looking at it in a test tube like that, they sometimes refer to the white blood cells as the buffy coat because it looks kind of like a fuzzy cotton ball or something like that in between. So here we have a centrifuge. Right, so you take and put them in the centrifuge and you spin them and you separate the cells from the plasma, and then you measure what percentage of that volume is taken up by cells. Right, so this would be the whatever percentage of that volume is, that would be the hematocrit. So here you take a blood sample out, phlebotomist takes a blood sample out, you put it into a test tube, you spin it in a centrifuge, and then you look at it, and the plasma takes up about 55% of the volume. Then we have the buffy coat, which if the cells are properly packed, which they weren't in that earlier picture, but if they're properly packed, the white blood cells usually take up less than 1% of the volume. And down here, the erythrocytes, the red blood cells at the bottom, generally take up about 45% of the volume. If you look at the constituents of the cells, right, so if you take a microliter of bload, a microliter of blood is a very small volume. It's like a microliter takes up about the same volume as the head of a pin, the fat side of a pin or the fat side of a nail. Okay, so if you take a microliter of blood, it will typically contain 5 to 6 million erythrocytes, and so erythrocytes are the largest constituent of what's called the formed elements, the cells. It'll have 250,000 to 450,000 thrombocytes or platelets that are involved in clotting the blood. And then the rest of the cells, 5,000 to 10,000, which is not a very large number, of the leukocytes. And those leukocytes consist of the lymphocytes and the granulocytes. All in all, the white blood cells take up about 1% of the volume of the blood, or less than 1%. Okay, so the hematocrit is around 45% of the blood volume. It's a little bit variable in women because they lose some blood cells once a month through menstruation. right? So of the 45% of the volume that's made up by the red blood cells, 99% of those, 99% of that 45% is erythrocytes, and less than 1% is leukocytes. Okay, for women, for instance, day one after menstruation, the percentage might be down to 35%, and then it works its way back up on day 20, just before menstruation. It works its way back up to 45, and then has to start over again. Okay, now if you lose blood because of an injury, think about this. If you lose blood because you have a cut, you lose blood, that's called hemorrhaging. So I'm going to ask you an academic question. If you lose, let's say you lose a liter of blood from your body, what do you think will happen to the hematocrit? Will it go up or down? You can pause the tape, you can pause the video for a minute and think about the answer and see if you got it right. Will it go up, down, or will it do nothing? Right. Okay, the answer is that it will do nothing, because you lost both cells and serum at the same time. So the fraction of the blood volume that's taken up by cells will remain the same. So if you lose blood due to hemorrhaging, you have a lower blood volume. Your blood pressure will probably drop, but you will not. The hematocrit will not change. Okay, now if they take you to the emergency room, one of the first things they do is to try and get your blood pressure back up, and they don't know your blood type, so they can't really put blood into you until they figure out what your blood type is. So as an emergency measure, they put in an intravenous tube. And then they pump you full of saline. Saline solution, of course, is just salty water, sodium and potassium chloride. And you replace, so let's say you lost a liter of blood because you were in a traffic accident of some sort. They take you to the emergency room and they put a liter of saline into you so that you have the same blood volume that you originally had. Now what happens to your hematocrit? Does it go up, down, or stay the same? Okay, so you can stop the tape, stop the video, and see what you think. Okay, ready for the answer? The answer is, of course, that the hematocrit will go down. So if you've lost blood and then they replace the volume by putting saline into you but not blood, then your hematocrit will go down. So you might have had 45% before the accident, and then they put the saline into you as an emergency measure to bring your blood pressure back up. Then the hematocrit could be down at 35 or even 25. So that's what they do in the emergency room. They want to make up the volume of blood as fast as possible, so they put an IV needle into you to replace the volume with saline. All right, let's look at the composition of blood. So the term formed elements, as I said before, that refers to cells. So another name for the blood cells is the formed elements of the blood. And they constitute about 37 to 45%. Men have generally 45%, and women can have somewhere between 37 and 45%, depending on what's going on with menstruation. And then the liquid portion of blood is called the plasma, right? And it constitutes about between 46 and 63% of the blood volume. Of the formed elements, 99% of the formed elements are erythrocytes, less than 1% are leukocytes, and less than 1% are platelets. If you look at the plasma, the liquid, it's 92% water, and then 7% of the volume of serum is made up by proteins, plasma proteins. Now, think about that. Can you think of any plasma proteins? All right, so on the final exam, I often ask, name 2 or 3 plasma proteins. And every year, students say hemoglobin, but hemoglobin is not a plasma protein because it's inside the cell. Right? It's inside the red blood cells, and so hemoglobin is not the answer. Right? But you've already heard of some other ones. You've heard of transferrin, you've heard of albumin. And then antibodies belong to a class of proteins called immunoglobulins. So the immunoglobulins are another class of serum proteins, right? So if you want to know about serum proteins, they include things like transferrin, immunoglobulins, and albumin, serum albumin. And then 1% of the volume of blood is taken up by other things like nutrients, lipids, carbohydrates, and gases. Okay, so the composition of the proteins, we said that 7% of the blood serum is made up of proteins. Of that 7% of serum that's made up of proteins, 60% is serum albumin, which transports things to the liver. It transports bilirubin, as we said before, and it also helps to regulate the oncotic blood pressure. Remember that the oncotic blood pressure is a kind of a reflection of how much blood is in the veins and arteries and the capillaries versus how much blood is in the interstitial spaces between cells in soft tissues. Okay, 35% of that 7% is made up of the globulins. The gamma globulins are the antibodies that are there to fight off infections, and antibodies are produced by B cells that have differentiated into plasma cells. We'll talk about that in detail later. And less than 1% of the proteins in serum are the hormones and cytokines and enzymes and other things like that, including the blood clotting factors, which we'll talk about very soon. Okay, now, speaking of blood clotting, 5%, we said that only 7% of the volume of blood is taken up by, or only 7% of the serum volume is taken up by proteins, and only 5% of those are fibrin, only 5% of those are a protein called fibrin, fibrinogen. And fibrinogen is an important protein in blood clotting. So we'll talk about blood clotting. All right, so let's talk about thrombocytes. And thrombocytes that are involved in blood clotting, which is, of course, the process called hemostasis. So, as we said, thrombocytes they are members of the myeloid lineage. They are fragments of a larger cell called a megakaryocyte that break off, and they cause the blood to form a clot, which is known as a thrombus. So you see where they get their name. A thrombocyte is involved in blood clotting, and you call a blood clot a thrombus. So if you have damage to the internal surface of a blood vessel, you make a blood clot, you make a thrombus in order to plug the hole. I don't know if you've ever had any of these really expensive automobile tires that contain chemicals. The chemical is a liquid as long as it's not exposed to air, but if you have a puncture in the tire, then the air gets in and the chemical becomes a solid and blocks the hole. Well, that's basically what thrombocytes do. They cause fibrinogen to form fibrin and so on, and then these fibers of fibrin kind of act as a net to rope in a bunch of red blood cells and block the damage, block the hole to the blood vessel from the inside. Okay, so hemostasis refers to blood clotting and blood vessel repair. Right, there are two soluble inactive proteins that are circulating in the blood that are just waiting to be activated by platelets, and platelets or thrombocytes will activate these two proteins in response to damage to the blood vessels. The two proteins are called prothrombin. The word pro, of course, means before. So prothrombin is activated by various blood clotting factors and turned from prothrombin into thrombin. And then active thrombin then turns around and converts fibrinogen. The word gen means to come from. It converts inactive fibrinogen into fibrin, which causes the blood clot to form. So the platelets do this by setting off a series of reactions, which is referred to as the fibrin synthesis cascade. A cascade refers to a waterfall where the water falls over the top of the mountain and then hits some rocks and bounces off and falls over those rocks and bounces off. So a cascade essentially means one enzyme activates another enzyme, which activates another enzyme, and ultimately you get this thing formed. So the fibrin synthesis cascade, it refers to a series of enzymes that are turned on in sequence, in a particular sequence, in order to cause the blood to clot. And ultimately they form active thrombin. Thrombin is a protein, active thrombin. Okay, so active thrombin converts inactive fibrinogen into fibrin, and it's actually fibrin that forms the clot. So fibrin fibers then tie together a bunch of clumps of red blood cells and white blood cells and lipids and thrombocytes and a bunch of other junk that forms a thrombus, or a blood clot, that plugs the hole on the inside of the blood vessel. Okay, so here we have a diagram of a damaged blood vessel. The platelets are able to sense that there's a hole here or a damaged part, and so they swarm to the area and they start an enzymatic cascade that causes thrombin to be activated, and then active thrombin causes fibrinogen to be converted to fibrin. Fibrin then makes these kind of rope-like structures that plug up the hole by bringing together a whole bunch of red blood cells. So these fiber-like structures here are the fibrin, and they're kind of acting like a net to catch a whole bunch of blood cells, red blood cells, and form a plug that plugs up this hole that we originally had in the blood vessel. So that is a thrombus. Here's a closer look at the thrombus. So here we have the fibrin fibers, right? These things, and they're kind of holding together a big clump of red blood cells that are blocking the hole on the inside of the blood vessel in order to stop the bleeding. Okay, so the fibrin synthesis cascade involves a sequential activation or a cascade of a bunch of inactive proteins that are referred to as the clotting factors. And the clotting factors are identified because you give them Roman numerals, and then you call them factor 1, factor 2, factor 3, and factor 4, and so on. Right, so two of the most important ones are blood clotting factor 8 and blood clotting factor 9. Somebody that has, we already mentioned the fact that there's a disease called hemophilia, which is a disease where your blood doesn't clot as quickly as it's supposed to. And people who have hemophilia have either a mutation to blood clotting factor 8 or blood clotting factor 9 that is stopping their blood from clotting properly. Right? So these clotting factors are involved in allowing the blood to clot. These clotting factors are produced by the liver. The liver is an old friend of ours, right? So there's another fancy thing that the liver does. I've said in previous lectures that the liver does a lot of things. The liver doesn't get any of the glory that the brain and the heart gets, but the liver actually does so many important things. And so often on final exams, I will say, give me a list of 8 or 10 things that the liver does. Just give me the list, and I'll give you 1 mark for each of those things on the list. And this is another thing you can add to the list. The liver makes the clotting factors, the factor 1, factor 2, factor 3, 4, 5, 6, 7, 8, 9, that allow the blood to clot. That's another thing the liver does. This clotting requires calcium and also vitamin K. Remember that we get vitamin K from our food, but also we get vitamin K from E. coli cells that are living in our colon. It's a symbiotic relationship. And as I mentioned earlier, hemophilia is a blood clotting disease, right, where these one or two of either factor 8 or factor 9 are mutated. Okay, so don't worry too much about this diagram. I don't expect you to remember it, but there are 2 ways that blood can clot. One of them is called the intrinsic pathway. That basically means this sequence of reactions that involves the blood clotting factors is set off in response to intrinsic damage to the blood vessel or internal damage. So a blood vessel gets broken from the inside and it sets off this cascade of blood clotting factors that ultimately leads to fibrin activation. On the other hand, if the blood vessel is damaged from the outside, it sets off a slightly different sequence of enzymes, but ultimately it ends up with active fibrin. Okay, so this is just showing you a close-up of the intrinsic pathway. You see all the blood clotting factors here, factor 12, factor 12-2, and so on, and all these other things versus the extrinsic pathway. But down here at the bottom of this chart, we have the common pathway. So the final common pathway, regardless of whether the blood vessel was damaged from the outside or the inside, the final pathway involves the creation of active fibrin, which then cross-links a whole bunch of blood cells, red blood cells, forming a clot or a thrombus. There's your thrombus. Okay. As I said, the intrinsic pathway simply means the blood vessel was damaged from the inside versus the extrinsic pathway damaged from the outside. The final common pathway is always the same. It activates fibrin, which ends up creating a clot called a thrombus. Okay, a little detail that we didn't discuss is that when a blood vessel is broken, either from damage from the inside or the outside, the smooth muscles that form the center layer of a blood vessel, will go into a spasm called a vascular spasm. And the vascular spasm is one of the things that helps to attract and activate the thrombocytes, vascular spasms. Okay, now here's another interesting thing about blood clotting. We said that endothelial cells are simple squamous epithelial cells that line the inside of a blood vessel. There are a lot of times when we find a simple squamous epithelium lining a particular surface, and we give it a different name instead of squamous epithelium. In this case, the endothelial cells, the simple squamous epithelium that lines the inside of a blood vessel, is coated on the inside with a chemical called heparin. And so heparin is present on the inner surface of the blood vessels. And when the platelets come along, the platelets are able to sense whether they are feeling heparin, and if they are, everything's okay. But if the thrombocyte contacts collagen instead, which you're not supposed to be able to feel, you're not supposed to be able to feel collagen on the inside of a blood vessel. So if the thrombocyte feels collagen instead of heparin, it assumes that the blood vessel is damaged, and thus the collagen layer, on the connective tissue on the outer layer of the blood vessel is exposed. The only way it could be exposed is if there was damage to the inside of the blood vessel. It starts activating the clot. Okay, so the interesting thing about that is that there are some people in the world who have problems with blood clotting. They have high blood pressure or something like that, and you don't want your blood to clot, right? So if you're trying to avoid a person having clotting blood, let's say they have a heart condition, they have occluded coronary arteries that can easily be blocked by a blood clot, you would give them a blood thinner that prevents or discourages the blood from clotting, and one of those is heparin. So sometimes you give people heparin to discourage the blood from clotting. Right, so here we have a damaged blood vessel, right, and then it starts out with a vascular spasm. The platelets will feel collagen instead of heparin on the inside, and they'll set off the clotting cascade. Clotting is not always good. So if the platelets and the fibrin form a clot, that's called a thrombus. It's supposed to stay in the area where the blood vessel is damaged, but it may not. So if the thrombus breaks loose and starts wandering around the bloodstream, we give it a new name and we call it an embolus. An embolus is capable of blocking blood vessels. So if the embolus wanders around and blocks blood vessels and cuts off the flow of blood, that is referred to as ischemia. Ischemia. Remember in biology, CH is usually pronounced K instead of CH. So this word is pronounced ischemia. So the word ischemia means that you've cut off blood to a certain area of the body. If you cut off the blood to a certain area of the body, then oxygen doesn't get through. So the lack of oxygen is referred to as hypoxia. Hypoxia. And if you deprive an area of blood for a long enough period of time, the tissue will die, and that's referred to as necrosis. Right, so you start out with a thrombus. If it breaks loose, you have an embolus that's wandering around the body. If the embolus blocks off a blood vessel, it's called ischemia. That leads to hypoxia, which can lead to necrosis or death of the tissues. The entire event, if that happens, is referred to as an embolism. Right? And the area of tissue that got killed as a result of the embolism is called an infarction. Therefore, what do you think a myocardial infarction is? Myocardial infarction, of course, is a dead part of the heart tissue because the heart was blocked. One of the coronary arteries was blocked by an embolus. You had an embolism that killed off a piece of the myocardium. Okay, so here we have a myocardial infarction. This piece of the heart here where the tissue is dead because one of the coronary arteries was blocked by an embolus, leading to ischemia, hypoxia, and then necrosis. Okay, how do you avoid embolisms? You have to prevent your blood from clotting, or at least clotting quickly. Okay, so there are a number of ways you can do that. Number one is don't eat trans unsaturated fats. Don't eat margarine. Don't eat chips and French fries that are fried in partially hydrogenated vegetable oil, because trans unsaturated fats are present in these types of oils. Regular oil, like canola oil, olive oil, those are natural oils. But in a lot of deep fat fryers that they use at fast food restaurants, they hydrogenate the oil. They bubble hydrogen through it, and that has the effect of breaking the double bonds in the unsaturated oil, but it also creates a lot of trans double bonds which don't occur naturally. So our body doesn't know how to handle trans unsaturated fats, and they usually end up just clogging up the arteries. So chicken McNuggets, margarine, chips, French fries, all of these things that are fried in partially hydrogenated vegetable oil encourage the formation of embolisms because they cause the arteries to occlude. An occluded artery means that it's narrower than it should be because the outside of, the inner part of the periphery of the artery is lined with junk, with this gunk called plaque, and unsaturated fats cause the formation of plaque in the arteries. So if you want to avoid embolisms, don't eat any of those fast foods that are deep fried in partially hydrogenated vegetable oil. Number two, don't eat too much salt, because salt has the effect of increasing the blood volume, which has the effect of increasing the blood pressure. The higher the velocity of the blood going through the body, the more likely the blood is to tear a thrombus away from the place that it was sitting. So a thrombus is sitting on top of a damaged blood vessel, and it's supposed to stay there if it's going to plug the leak. But if the blood pressure is too high, the blood pressure may just sweep the thrombus away, and then the body has to build a new one. But the one that the blood pressure swept away is going to travel around and cause ischemia to some other part of the body. Another way to avoid embolisms is to do regular cardiovascular exercise, because that makes your blood vessels more flexible. And the more flexible they are, the better they are to respond to high blood pressure. And finally, there's something else that everybody can do. These drugs are common painkillers. These are the generic names for these drugs, and these are the brand names for these drugs. Right, so ibuprofen goes by the brand name Tylenol. Acetylsalicylic acid goes by the brand name aspirin. And ibuprofen goes by the brand name Advil, but it's the same thing. You can take a small amount, a low dose of one of these drugs every night, and it has the effect of preventing thrombuses from forming. It has the effect of discouraging the blood from clotting. Do you know which one? You've probably heard it in the news, right? Did you guess correctly? It is ASA, acetylsalicylic acid, or low-dose aspirin. Notice that if you have a headache, you take maybe 250 milligrams of aspirin, or 500 if you have a really bad headache. But if you take a low dose, 81 milligrams every day, that discourages blood clots from forming, and it could, it has a significant impact on your life. It significantly lowers the probability that you will have a heart attack from an embolism, because it prevents or it discourages the blood from clotting. So people, especially older people, middle-aged and up, age late 30s and then on up for the rest of your life, you should take low-dose acetylsalicylic acid every day. So you take 81 milligrams of aspirin every night before going to bed. All right, let's do a little side story. When we were talking about the blood vessels the other day, and we were talking about the profession of phlebotomy, I showed you a vacutainer that had a light blue cap, and I said this is to do something called the PT test. PT test stands for prothrombin. It's a test that measures how fast your blood clots, and if your blood clots too quickly or too slowly, it might mean that you have too much fibrin or too little. Why would you have too little fibrinogen floating around in your bloodstream? Well, maybe it's because all of the fibrinogen is being used someplace else in the body. Right? So that would tend to slow down your blood from clotting, because the fibrinogen is being used for maybe you have an internal injury and you're having some internal bleeding or something like that. So it is useful to measure how fast the blood can clot. And you do that with something called the PT test, or the prothrombin test. Okay, so the PT test, how long does it take for your blood to clot? If it clots too fast, it indicates there's a lot of that a lot of the fibrin in your body has been activated, and then you ask why. Well, no, there's something wrong, and you should look further into why. On the other hand, if the blood clots too slowly, it's because all of the fibrin has been activated and is actually being used to make clots somewhere else in the body. And again, you would ask yourself, why? What's going on? Why did that happen? And one possibility is deep vein thrombosis. So we talked about deep vein thrombosis the other day when we talked about the blood vessels. This is where you have kind of deep veins in the legs that have become stretched and bent out of shape, and so you have these pools of blood that are kind of collecting in these stretched veins, and blood doesn't like to sit in the same place for very long, and if it does, it tends to form clots, and so that gives you deep vein thrombosis. These clots can then wander around and plug different parts of your body and cause real problems. So you can tell if somebody possibly has deep vein thrombosis because they have an extended PT time. Okay, so how do you do the prothrombin test, PT test? Okay, so the phlebotomist comes around and they take the light blue capped tube, and that tube contains a buffer called sodium citrate, which actually inhibits blood clotting. It stops it. So it's the light blue tube. And then once you've got the tube, you add calcium. Actually what you do is you centrifuge the blood first and then transfer the serum to another tube, right? So you just have the liquid part of the blood. And then you add calcium, and the calcium has the effect of turning off the inhibitory effects of the sodium citrate. So now your blood is allowed to clot, and you measure how long it takes to clot. Okay, then you measure the number of seconds it takes for the blood to clot by shaking the tube and looking at it. When you see a clot in the bottom, you stop your stopwatch. And on average, it should take about 12 seconds for the blood to clot. Okay, now you compare that to a control sample of normal serum. Right, so you have two test tubes. One of them contains your patient's serum. Another one contains a controlled sample. You add calcium to both tubes, and therefore you set off the clotting reaction. And then you time each of those tubes, see how long they take to clot. And then you divide the time that it takes the patient's blood to clot. You divide that time by the amount of time it takes the control sample of normal serum to clot. And that gives you what's known as the international normalized ratio, the INR. Right? So I could ask you on the final what the INR is. Which test is it part of? It's part of the PT test. And what does it mean? It tells you whether the blood takes longer or shorter, a longer period of time or a shorter period of time to clot. Right? So normally, if your blood is normal, the INR should be 0.9 to 1.1. Okay, I'll ask you a question. If the INR ratio is 0.5, does that mean that the patient's blood is clotting faster or slower than normal? Right? Of course, it's clotting faster than normal. Right? What if the INR is 1.5? Is the patient's blood clotting faster or slower than normal? Think about that. All right, we talked about what the PT test is for. The other tubes, we talked about what they are for. All right, now something that we've all heard of, the ABO blood grouping test, which it means that when a patient loses blood, you have to transfer blood from a donor into the recipient to replace the blood. We've been doing this for 100 years. And we know that you can't transplant blood from anybody into anybody else. In fact, you can only transfer certain types of blood into certain types of recipients based on their blood type. And there are three types of blood, actually four types of blood: type A, type B, type O, and type AB. And we'll discuss what that means and discuss who can donate blood to who. All right. If you put a foreign protein into your body, that means a protein that you do not normally have, your immune system will perceive of it as being a foreign invader and will destroy it by attacking it with antibodies. Right? So your immune system will destroy foreign invaders by generating antibodies. So any protein, any foreign protein that you do not have, it's not recognized as, your immune system says that's not part of me. It must be part of a hostile invader, and it provokes an antibody reaction. Any foreign protein in your body that provokes the formation of antibodies is referred to as an antigen. An antigen. That word will be more important when we discuss the immune system, but for now, an antigen, if you have antigens on your red blood cells, and then you transplant your red blood cells into somebody else who doesn't have that antigen on their red blood cells, their immune system will destroy it because it perceives it as an invader. Okay, now there is in fact a special type of a protein that you find on the surface of a red blood cell that's a special type of protein that's called a ceramide. We're not actually going to discuss what ceramides are in this course, but it's a special type, slightly unusual type of a protein that is found on the surface of red blood cells. And there are two different types of this ceramide antigen, and they are both encoded by the same gene. Right? So remember that we talked about if you have two slightly different versions of a gene that give you two slightly different versions of the same protein, those two versions of the gene are referred to as alleles. Right? So there are two alleles, there are actually three alleles of this particular ceramide, right? And they are referred to as the A allele, the B allele, and the O allele. And we'll talk about what that means in a second. But if you take blood from one person who's called a donor and you put it into another person who's called a recipient, that's called a transfusion. If the donor's red blood cells have a different ceramide on the surface than the recipient's immune system will attack and destroy their red blood cells. So here we have the blood types. This is blood type A, blood type B, blood type AB, and blood type O. Right? So if you take blood cells from a person who has type B blood, a donor, and you transfuse them into a person who has type A blood, the type A person's immune system will destroy those blood cells, and you may as well not have bothered to put them in the first place because they'll just be destroyed. Okay, that's why we sort blood according to blood type. So you have blood type B, blood type A, blood type AB, and blood type O. Some of you may have noticed that there's a little marking here that says Rh positive and Rh negative on some of these bags of blood. And we'll talk about what that means in a minute. That's related to another antigen on the surface of red blood cells called the rhesus factor. All right, so blood type, how does the blood type work? So again, if you have two different alleles, we have actually three different alleles of the gene that makes the ceramide on the surface of the blood cells. Right, so a slightly different version of a gene is referred to an allele. That's why some of us have brown eyes and some of us have blue eyes. Okay, so the A allele codes for a certain ceramide protein. The B allele codes for a slightly different ceramide protein, and the O allele is what geneticists call a null mutation, which means that it doesn't produce any ceramide at all. Right? So we have two alleles that are slightly different that produce a slightly different ceramide protein, and we have a third version of that gene that's basically a nothing mutation that doesn't produce any protein at all. Right? That's the O allele. Humans, of course, are diploid, meaning we have we can have a maximum of two different alleles for every gene. So we have two copies of each gene. If you have two copies of the A allele, you call a person like that a homozygote with respect to the blood type antigen. But remember that we have two different copies of the gene, but they're both the same. So technically, you would say that we only have one. A person like that only has one allele. A person who has two copies of the B allele, again, there's only one allele present. You have two copies of it, so you would say that that person has only one allele, and they are a homozygote. But what if a person has one of the B alleles and one of the O alleles? Then they would have one of each. They would have two different alleles. You call a person like that a heterozygote, and there are two alleles present. So when you're doing your genetics class, which is very likely, just remember that when you have the same two copies of the same allele, a real geneticist, a proper geneticist, would get upset if you said, "Oh, there are two alleles present." No, there's only one allele present. You have two copies of the same allele. All right, so it is these alleles that are determining what antigens are on the surface of the red blood cell. Okay, so somebody who has the genotype, remember the genotype is all the genes that you have, and so we take into account the fact that humans are diploid. So if a person has the AA genotype, that means they have the A antigen on their red blood cells and they have blood type A. Somebody has two copies of the B antigen, two copies of the B antigen, they have only the B allele, they have only the B antigen on the blood cells. Two copies of the O allele means that they have neither A nor B on the surface, they have nothing. And then you get into these, right? So if you have the A allele and the O allele, they're still blood type O because they only have the A antigen on the surface. If they have B and O, that means they're blood type B because they have only the B antigen on the surface. And if they have one of each, they have one of each of the A allele and the B allele, that means they have both antigens on the surface, right? So they are blood type AB, just like this, right? And that determines, these gene alleles determine which version of the ceramide, or both in this case, or neither, are present on the surface of the blood cells. So this is the AA homozygote or AO heterozygote gives you blood type A. BB or BO gives you blood type B. AB or AB gives you heterozygote AB gives you blood type B, and OO gives you neither, gives you blood type O, blood type O. Now, if you happen to be an A type, if you happen to have the A blood type, that means that your body contains antibodies that will destroy the B antigen, which you see over here. Right? So that is why you can't take B blood cells and transplant them into an A person, because their B antibodies will destroy the B blood cells. Right? And the reverse is true for somebody with blood type B. They have antibodies that are there specifically to destroy the A antigens if they come inside the body. Now this is interesting. The person who has AB blood type has both antigens, both types of ceramide on their surface, and they have antibodies to neither, because remember, you have antibodies in your system that are there to destroy proteins that are foreign to you. Right? So if this person has both A's and B's on their blood cells, then those are not foreign proteins, and so they don't have any antibodies that would destroy either one. A person with type O blood has neither of those ceramide proteins on the surface, and so therefore they would destroy both of them. Right? So you cannot transplant any of these blood types into a type O person because their antibodies would destroy them. All right, let's look at genotypes and phenotypes one more time. The genotype is the genes you have. The phenotype is how those genes express themselves on the outside of the body. What do they look like when they're put into action? What do they look like when they produce proteins? That's the phenotype. Okay, so AA and AO give you blood type A. BB and BO give you blood type B. AB gives you blood type AB, and OO gives you blood type O. All right, how do you go about determining which blood type somebody has? You use a test which is known as the agglutination test. The word agglutination is what happens when you mix antibodies that react with blood cells that they react to. So let's have a look here. You put a few drops of blood, usually 2 drops of blood, onto a glass slide, and then you mix 2 different antibodies with those drops of blood. Right, so you mix antibodies that attack the A antigen with one of the drops of blood, and then you mix antibodies that attack the B antigen with the other drop, and then you look to see which one of those 2 drops will agglutinate. Agglutination means the blood cells kind of clump in preparation to being destroyed, and so that agglutination is quite obvious. Right, so let's take a look here. Here we have a slide. Right, now here we have a person, we put 2 drops of blood on the slide, we put 2 drops of their blood on the slide, and then we mix antibodies that react with or attack the A antigen into this drop, and you see agglutination. That's agglutination. For the other drop, we put in antibodies that attack the B antigen, and they don't that attack the B antigen, and doesn't do anything. It doesn't cause the blood to agglutinate. And therefore, because the A antibodies caused the blood to agglutinate and the B antibodies did not, that person must be blood type A. Right, so this would make a very good final exam exercise to show you a picture like this and then get you to tell me every blood type of all of these people. Right, here's another example. We take 2 blood drops from the same person, and then we add antibodies that react with the A antigen here. Nothing happens. We add antibodies that react with the B antigen here, and the blood agglutinates. Therefore, this person must be blood type B. Okay, what about this person? We take 2 drops of their blood, and the drops of blood react with both of the antibodies. That's because they have both of those foreign antigens on they have both of those antigens on their surface, and they must be type AB. And then over here we have the antibodies react with neither drop of blood. Therefore, these red blood cells have neither ceramide on their surface, and this must be blood type O. This is the actual tray, a little plastic tray that pharmaceutical companies will sell you to do the actual blood test. So you put 2 drops of blood into this little tray, and then you mix each of the blood drops with either the anti-A or the anti-B antibodies, and then you look for agglutination. You notice that there's a third well here, which is where you add antibodies for another blood surface factor called rhesus factor, and we'll talk about that next. So people are generally of the blood type A, blood type B, blood type AB, or blood type O. And then in addition to that, we have to find out whether they are Rh positive or Rh negative, and we'll talk about that next. Before we do that, however, let's just take a little look at who can donate blood to whom and who can accept blood from whom. Okay, so here we have a bunch of blood donors on the left, and we're going to take blood from them and transplant them into a recipient, and you tell me which ones are compatible. So go through this exercise on your own time, but basically that, of course, is compatible because you have the same blood type. I'll give you a minute if you want to pause the video here, and then run the video again, and I'll give you the answers. Okay, so this one, blood type A, would also be compatible. Blood type B would not, right? Because this person here does not have the B antigen on their blood cells, and so the blood type B would be destroyed if you put it into this recipient. The BB genotype, giving you the type B blood, would be destroyed as well. Okay, now what about this? This is a type AB person. Okay, so the blood type AB, this person has the A antigen on their blood, which is okay for the recipient because the recipient also has the A antigen. And so they wouldn't perceive the A antigen as a foreign invading protein, a foreign invading ceramide. But they also have the B antigen, which the recipient does not have. So their immune system would destroy the blood cells that have the B ceramide, and the fact that they have the A ceramide as well doesn't matter. All right, now what about this person, type O? Right, this person, you could put them in there because their blood cells have no ceramides on the surface, so there's nothing there that this person's immune system would react with. All right. Now let's try it for a different recipient. You tell me who can donate and who cannot. I'll give you a minute, pause it, and then see if you got the answer. The answers are yes, yes, no, 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 because of this, and yes. Okay, try this one. No, no, yes, yes, no, yes. Try this one. The answers are yes, 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 yes. Okay, try this one. No, 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 no. Yes. Okay, so a person who has type AB blood can receive blood from any of these other people, but they cannot donate blood to anybody except another AB patient. And because they can receive blood from anybody without their immune system rejecting it, they're referred to as universal recipients. Okay, a type O person can only receive blood from another type O person, but they can donate blood to anybody without fear of it being rejected, and so we call people with blood type O universal donors. All right, this is a little anecdote which you may have heard of a few years ago. The Duke University is a university in the United States. They have one of the best cardiology divisions in the world. They specialize in pediatric cardiology, so that's transplanting hearts into children. And a few years ago, one of their top surgeons, this guy over here, made a mistake and transplanted a type A heart into a type O patient and killed her, basically. And this is the head of the cardiology department explaining to the reporters why it wasn't really his fault, because it's not the surgeon's job to check the blood type of the heart, which is true. It's the blood granting. It's the blood or the organ donor agency whose job it is to check the blood type of the organ that's being donated. But it was a big disaster, right? It was a very fundamental, simple mistake, which anyone, even first year high school and college students, know that you cannot transplant a type A heart into a type O recipient because of the reasons we just explained. Okay, now what? Now there's another interesting thing. What if a mother is type O blood and she's carrying a type A baby because the man that she conceived the child with, her husband or boyfriend or whatever, he had blood type A, right? So now you have a woman who has type O blood who's carrying a baby that's type A has type A blood. Will the mother's antibodies destroy the baby's blood cells? Okay, so let's see what happens in that case. What happens if a mother's carrying a baby with an incompatible blood type? Right, so let's say you have a father who's genotype AA, so he has blood type A, and a mother whose genotype is OO, so then they will definitely have a baby that has blood type A, genotype AO. Now, what happens usually is that the placenta is how the baby gets oxygen. And so the baby's lungs usually up to right before birth, the baby's lungs don't actually work and they're certainly not inhaling any air in the womb. So the child's blood gets oxygenated through a structure called the placenta. And so the placenta, what happens in the placenta is that the mother's blood vessels and the baby's blood vessels come very close together. So close, in fact, that they can exchange oxygen and gases and nutrients. But the blood cells do not actually touch. Now, in order to mount an immune reaction, and we'll talk about this later when we discuss the immune system, but in order to mount an immune reaction, the white blood cells in mother's blood have to make contact with the baby's red blood cells before you'll set off an immune reaction. The placenta prevents that from happening. Antibodies can cross the placenta, but the blood cells themselves do not cross. The blood of mother and child do not mix. Right? So the mother can't make an immune system reaction to a baby's blood unless her blood directly mixed with the baby's blood. Okay, so on the right we see a placenta. You know the placenta, of course, is attached to the baby's navel, the umbilical cord, through the umbilical cord. Remember, the umbilical cord is a source of hematopoietic stem cells, as we discussed before. Right? And within the placenta, the mother's blood vessels and the baby's blood vessels come very close together, but they don't actually touch. They just come close enough together to allow gases to perfuse across the walls. But the blood doesn't actually touch. So an incompatible blood type of a baby should be a problem. So during the first birth, first time, let's say you have an AA man and an OO woman, and she's carrying a type AO baby. The first time she gives birth, that won't happen because the baby's blood and the mother's blood don't mix. But sometimes during delivery, the placenta tears so that the mother's blood and the baby's blood mix, and therefore the mother will start to mount an immune reaction to the baby's blood. She will make antibodies that will destroy the baby's blood. But that's not a problem because the baby just came out. Right? So the first baby got away with that. But during the birth, the placenta rips and the baby's blood and the mother's blood get mixed a little bit. That means that the next time they have a baby, there could be a problem because the mother's immune system has already made antibodies that will destroy the baby's blood. Right? Usually the reaction between a type O mother's blood and a type A or a type B baby's blood is not that severe. It doesn't cause a serious problem. We also know that there's a thing called the rhesus factor on the surface of the blood. The rhesus factor, if a woman who is rhesus factor negative is carrying the child of a man who is rhesus factor positive, so the baby is rhesus factor positive, that can cause a problem. And so there are various drugs that the mother can take to suppress the production of antibodies that will damage the baby's blood. Okay, but anyway, we heard that, so that's the rhesus factor. We also know that maybe the ABO incompatibility might cause a little bit of a problem as well, but it's usually not a severe problem. It's usually not a big problem, unlike the rhesus factor, which usually is. Okay, so but just for fun, let's talk about whether these men and these women are compatible in terms of making a baby. Could there possibly be a problem? Okay, so here we have a man and a woman with the following genotypes, and then here we have they're going to produce a baby, and you tell me whether the baby's blood will be compatible with the mother's blood or not. Right? So, what are the odds that the baby, first of all, what are the odds that the baby will have an incompatible blood type? Okay, so the mother only has the two O alleles, so the mother is guaranteed to donate an O. Right? So we already know half of what the baby's genotype is going to be. The father has an O, so 50% of the time he will donate an O in his sperm, right? So then in that case, the baby will be OO, and there's no problem. There's no incompatibility problem with the mother. But half the time he will donate the A allele in his sperm cell, right? And the baby will be blood type AO, will be genotype AO, which is blood type A. Right? So that could possibly cause a problem. Right? So is there possibly a problem here? What are the odds that, let's say that this child has already, this couple has already had a child that has blood type A. What are the odds that their second baby will also have blood type A? Well, the odds are actually 50% because the father donates the A antigen to the baby 50% of the time. Right? So there could be a slight problem with that. What about this? Okay, so the mother is going to donate one of her O alleles to the baby, and the father is going to donate the B allele half the time or the A allele half the time, right? So either way, the baby will have an incompatible blood type. Okay, so let's say that the first time, their first child, the baby has a B blood type, the mother's blood doesn't mix with it until it's born. Then next time, what are the odds that their second baby will be a B and there'll be a problem versus their second child being blood type A? Well, the odds of their second child being blood type B are 50% because he donates the B allele half the time. What about this one? Okay, so if the mother is blood type BB, if the father donates the A allele twice in a row, there could be a problem with that, with the second child. If he donates the O both times, or if he donates the O the first time and the A the second time, then there's not generally going to be a problem. Okay, what about this? Okay, what do you think about this? I'll let you stop the video for a minute and see what you think. The answer is, in fact, this could never be a problem because the mother has both the A and the B, so no matter what the father does, her blood is not going to reject the baby's blood because the baby's blood will always have less antigens and less antigens on the surface than hers does, and it will always be the same antigens. All right, what about the rhesus factor? Okay, the rhesus factor is the immune system reacts very strongly to the rhesus factor. The word rhesus factor, it's abbreviated RH. It's just another ceramide on the surface, and it was originally discovered in rhesus monkeys. So rhesus monkeys are a type of monkey, and they first discovered this protein on the surface of monkey blood cells, but we have it as well, so they call it the rhesus factor or the Rh factor. All right, you either have it or you don't, and there are two possible alleles, right? So any of these combinations, if you have two of the dominant allele, then you'll be Rh positive. If you have one copy of the Rh plus and one copy of the Rh minus allele, you'll still be Rh positive. It's sort of like the A's and the O's and the B's that we saw before. But if you have two of the recessive alleles, then you're Rh negative. Okay, so what happens if you have an Rh positive mother carrying an Rh minus baby? That's not a problem. But what if you have an Rh minus mother carrying an Rh positive baby? That could be a problem. So in fact, if an Rh minus mother is carrying an Rh positive baby for the second time, you get something that's referred to as HDN. And I could ask you about this on the final exam. I could say, what does HDN stand for? It stands for hemolytic diseases of the newborn. All right, so in the old days, they used to make people, before they would allow people to get married, they would and start having children, they would insist that they get a marriage license, specifically so that a doctor could look at the marriage license and say, okay, in order to get a marriage license, you had to have a blood test. And the reason why they wanted to make sure you had a blood test before you had children with your spouse was because they wanted to check if the baby could possibly be in danger of hemolytic diseases of the newborn. So they want to know whether the mother and the father, what are their blood types? Is it A, B, or O? And they wanted to know what are the mother and father's status with regards to rhesus factor. So that is the mother Rh positive or negative? Is the father Rh positive or negative? Right, so that was the idea behind getting a marriage, behind forcing people to get a blood test before they were allowed to get married and have children. These days, people don't necessarily get married to have children anymore. But it's still a good idea to get a blood test to tell if they are compatible. In fact, that was the purpose of the marriage license in the first place. But as I said, people may not get married these days to have children. But if they're planning to have children, they should always check to see if their blood types are compatible or if there's a problem. Okay, so if the first baby is Rh positive and is being born to an Rh negative mother, it's not a problem. But the second one, the mother's immune system has been what's known as sensitized. It's been exposed to the Rh antigen, and the second time the immune system is ready for it, and it will attack the baby's blood while the baby is still in the womb, because the antibodies can pass through the placenta, and the baby comes out looking a little bit blue. So this is known as a blue baby syndrome, which is the technical term for it is hemolytic diseases of the newborn. And this is one of two blue baby syndromes that we learned about in this course, right? The other blue baby syndrome is when a baby comes out blue because they have a ventricular septal defect and a reduced pulmonary artery, and that of course is called tetralogy of Fallot, right? So if I ask you for the two different pathologies that cause a baby to be born blue, you would say number one is tetralogy of Fallot, which is caused by a circulatory problem, and the other one is caused by hemolytic diseases of the newborn, which is where you have an incompatible blood type, particularly the rhesus factor. So this would be a rhesus factor, an Rh minus mother's second child with an Rh positive father, second time for an Rh positive baby, and then the baby comes out looking blue because the mother's immune system has destroyed the baby's blood. And so when the blood is poor in oxygen, it makes you appear a little bit blue. All right, so to avoid this, the mother can take a drug called RhoGAM. And again, that's another final exam question. I would ask you, what is the drug RhoGAM for? And the drug RhoGAM is used to suppress the production of antibodies by the mother so that her immune system won't attack the baby as it's being developed. All right, with the little time we have left, let's just talk about some common blood pathologies. Kind of interesting stuff. All right, anemia specifically refers to the lack of red blood cells alone. Leukopenia refers to the lack of white blood cells. Leuko means white, penia means lack of, right? So leukopenia means lack of white blood cells. Thrombocytopenia means lack of thrombocytes. And pancytopenia means lack of all blood cells. The term pan means everything, right? So When you talk about an epidemic, you're talking about a disease that's restricted to a certain part of the world, and when you talk about a pandemic, you're talking about a disease that spreads to the entire world. So the word the prefix pan means everything, right? So pancytopenia, we discussed in the first week of lecture that cyto refers to cells, penia refers to lack of, and pan means all of them, everything, right? So pancytopenia is a lack of all blood cells, both red and white. So those are the terms at the top of the list refer to lack of cells. And then down at the bottom, I just introduced three terms that refer to an excess of cells. So pathologies that are caused by having too many cells. So polycythemia is the pathology of the blood where you naturally have too many red blood cells. Leukemia is where the white blood cells, particularly the circulating white blood cells, the B cells and the T cells that circulate in the blood, they proliferate uncontrollably because one of them has developed cancer. And then hemophilia A and B is actually a lack of clotting. Sorry, it's a lack of clotting factors, and it causes the blood to clot more slowly. Okay, so let's look at them one at a time. These are too few cells, right? So this is normal blood, right? And then down here on the bottom, you see anemic blood that has fewer red blood cells. It's a little bit difficult to tell whether they're too many or too few because it has a lot to do with how you prepared the slide. You have to be a real expert at making a slide and then looking at it to see what it means. But anyway, that's kind of what it looks like. And then polycythemia, right? Here's polycythemia vera, which is a form of polycythemia. You can see there are way too many blood cells. So that's probably what Lance Armstrong's blood looks like after he's been blood doping, after he's been injecting himself with erythropoietin to win bicycle races. And then here, this is a normal blood smear, so you can see, it's called a smear because you put a drop of blood on a glass slide and then you smear it across so that the blood cells are not sitting on top of each other, blocking each other from view. And then leukemia, so Lance Armstrong. All right, leukemia. So this is the normal ratio. This is the normal ratio of white blood cells in blood. They only constitute about 1% of the cells in blood. But if you have leukemia, they're up there at about 50% or whatever of the hematocrit, and that's bad because they start destroying things inside your body. So that's what happens with leukemia. You have an excess of white blood cells. Okay, so anemia. Now, why do you have a lack of erythrocytes in the first place? Okay, so one reason is that you don't make enough of them, right? So you don't make enough of them to begin with, or you don't have enough of them because your spleen is destroying them when it's not supposed to. And those particular types of anemias are referred to as autohemolytic anemias. Auto, meaning yourself, and then hemolytic. The term lytic, of course, means to destroy or tear apart, and then hemo refers to the blood. So you automatically destroy your own blood cells. It's an autohemolytic anemia, right? So let's discuss examples of each. Okay, iron deficiency anemia. Remember we said that hemoglobin contains iron. And if you don't have enough iron in your diet, for instance, then your body can't make enough hemoglobin. If it can't make enough hemoglobin, it says, what's the point of making all of these red blood cells when I don't even have enough hemoglobin to put inside them? So the body, if you don't have enough iron in your body, usually because you have an iron deficiency diet, your body cuts back on the amount of red blood cells that it produces because it doesn't have enough hemoglobin to put into them anyway. In addition to that, it cuts back on the number of red blood cells it makes, and the red blood cells that it does make, they look a little bit empty. They look a little bit pathetic because they don't contain as much hemoglobin as they otherwise would. And the consequence of that is that they're even emptier than they usually are, and the zone of central pallor looks bigger because the blood cells are kind of empty. All right, sickle cell anemia is caused by a genetic mutation, a specific genetic mutation that's more common in African people of African descent. This mutation survived in the population because if you live in Africa where you have malaria, having sickle cell anemia gives you some protection against malaria. So there are many examples of mutations that are bad on the one hand, but they give you a benefit on the other hand. And so sickle cell anemia is an example of that. If you live in an area where you don't have mosquitoes and malaria, then there's no reason sickle cell anemia is 100% bad, right? But if you live in a place where you do have malaria, it's handy. So what actually happens in sickle cell anemia is that there's a point mutation, a single nucleotide change in the globin gene, which causes the globin to function improperly, so that the globin that's produced switches from being a liquid soluble protein to an insoluble protein under conditions of low oxygen. And then when hemoglobin is dissolved in water, as it is in the blood, it takes up more space than it does when it's a solid. So when the blood cells are traveling to areas of the body where there's not much oxygen, the hemoglobin will turn from a liquid into a solid, taking up less volume, and the cells will collapse and form a sickle shape. And the spleen interprets that sickle shape as being a worn out blood cell. The blood cell might have just been created an hour ago, but the spleen will destroy it because it thinks it's an old worn out blood cell. So that is an example of an autolytic anemia. Spherocytosis is a genetic mutation that causes the cell membrane to become weak, allowing more water to diffuse into the cell, causing the cell to swell up. And again, the surface of the blood cell doesn't feel proper, so the spleen will destroy those cells. And so spherocytosis is another example of an autolytic anemia. Okay, vitamin deficiency anemia, you don't have enough vitamin B12 and it causes the cells to be malformed. And here's an interesting one called thalassemia. Thalasia is a Greek word, an ancient Greek word meaning the Mediterranean Sea. And this disease is called thalassemia because it is more common in people who live in the Mediterranean, specifically Greeks, people who live in Greece, Crete, or Italy. Will more commonly have a higher chance of having thalassemia. It's a genetic mutation that leads to a straight out lack of production of red blood cells for reasons that we don't quite understand. Okay, let's look at iron deficiency anemia. There's not enough iron in your diet to make hemoglobin, so why do you make so many cells if you don't even have enough hemoglobin to put into them? The consequence of this is that even the cells that you do make contain less hemoglobin than they otherwise would, and they have an enlarged zone of central pallor. So these are hemolytic. These are this is an example of iron deficiency anemia. This is normal blood. You see that the zone of central pallor takes up about a third of the cell volume. This is iron deficiency anemia. Notice that the zone of central pallor is at least twice as big. It's not true for every cell, but it's true for most of the cells, right? So this is by looking at that, a hematologist would be able to look at that blood and say that's that's iron deficiency anemia. All right, sickle cell anemia, you have a mutation to the gene. It's inherited, and then it causes the cells to form a sickle shape in certain parts of the body, and then the spleen will destroy those cells, autohemolytic anemia. Okay, so here's normal blood over here, and then on the right we have a very characteristic feature, sickle cell shaped blood cells. Spherocytosis, genetic mutation that causes the cell to become weaker. More water can diffuse inside through osmosis. The cell swells up and the zone of central pallor disappears. And the spleen senses that these cells are not the right shape and it destroys them. So again, this is an example of autolytic anemia. So here we have normal blood on the left, the zone of central pallor taking up 1/3 of the cell. Here we have spherocytosis on the right. You can't see the zone of central pallor at all because the cell is now completely round because it's absorbed so much water. Okay, so here's an exercise. You tell me whether the blood is normal or anemic. In this case, you can stop the video and then see if you can guess, and then I'll tell you the answer, and you can see if you got it right. Okay, what do you think about this? Answer, I would say this is normal blood. The zone of central pallor takes up about a third of the cell volume, and none of the cells are an abnormal shape. do you think about this? Answer, this is quite clearly iron deficiency anemia. How about this? Obviously, this is sickle cell anemia because that's the only anemia where you have these sickle-shaped cells. What about this? This is spherocytosis because the zone of central pallor has disappeared. Notice this nice, beautiful B cell or T cell. This is a nice, beautiful lymphocyte there. What about this? Answer is that this one is a very difficult one to call, but I think this is, a few of the blood cells look weird, like this could be an example of spherocytosis, but I think it's not. Most of the blood cells look normal, and by the way, I believe that's a megakaryocyte. I'm not an expert on hematology, but I believe that's a megakaryocyte. What about here? Notice that some of the cells have a normal zone of central pallor and some of them have an enlarged zone of central pallor. So this again is kind of a judgment call that would call for an expert, a hematologist, somebody who looks at blood for a living. But I think this is probably iron deficiency anemia. What about this? This is our old slide that I showed about an hour ago that shows the normal. This is, what do you think? This is quite obviously iron deficiency anemia. Obviously sickle cell anemia. Perhaps polycythemia, but it depends very heavily on how you prepared the slide. Probably a few of the cells look normal, but most of them look kind of empty, so that would probably be iron deficiency anemia. All right, what about hemophilia? Final thing we'll discuss, I believe. This is a genetic mutation to one of the blood clotting factors, either factor 8 or factor 9, that are produced by the liver. This is something that's known as an X-linked recessive trait, which means that if you're a diploid person, you have two copies of the gene. If one of the gene is the mutant version and the other one is normal, the normal one can kind of mask the presence or disguise the presence of, or hide. It can hide the fact that you have one mutant gene, and so your blood clots normally. However, the gene is located on the X chromosome. The locus for the gene is located on the X chromosome, which means that if you're a woman, you have two copies. If you're a man, you only have one. Males only have one X chromosome. So if you are carrying the hemophilia mutation, if you're a man, you stand a much greater chance of having hemophilia. You have a much greater chance of showing the fact because you only have one gene, and if it's mutant, then you're a hemophiliac. If you have two genes and only one of them is a mutant, then you're not a hemophiliac. So this hemophilia is a mutation to one of the blood clotting factors, either factor 8 or factor 9. Those genes are carried on the X chromosome. Therefore, there are probably about 10 times as many male hemophiliacs as there are female hemophiliacs. Women are considered to be carriers. Okay, so because they may have a mutant version of the clotting factor, but they don't have hemophilia. If they mate with a man, and then they remember that sons get their X chromosome exclusively from their mother. So a woman who is carrying a hemophilia mutant gene has a 50% chance, all of her sons have a 50% chance of being hemophiliacs if a woman is a carrier of hemophilia. All right, now hemophilia A is what you call it if you have a mutation to blood clotting factor 8, and hemophilia B is what you call it if you have a mutation to blood clotting factor 9. Right, this is just demonstrating how an X-linked recessive disease with a carrier mother works. If the mother has, in this case, the blue means the mutant gene, right? So this mother is a carrier. The father has the orange allele, which is the normal one. So the father is not hemophiliac. The mother is not hemophiliac, but she is a carrier. And if you look at the sons, half of the sons will get hemophilia and half the sons won't, because the sons always get their X chromosome from their mother. All right. Just a little anecdote. Hemophilia is spread throughout all of the royal families of Europe, and it initiated with Queen Victoria, who had lots and lots of kids and lots of grandkids. So all of the royal families of Europe kings of Norway, king of Spain, king of England. They're all related to Queen of England. Of course, they're all related to Queen Victoria, who was the progenitor, the first one to was believed to be this one who initially spread around the hemophilia mutant gene. All right, so just talking about blood today, we talked about what hematopoiesis is and how blood is formed in the flat bones. We talked about the life cycle of a erythrocyte. We talked about the two blood cell lineages. You're definitely responsible for knowing which cells belong in which lineage. We learned about blood cell compatibilities. We learned about the hematocrit. We learned about hemostasis and blood clotting, and we learned about some of the more common blood cell pathologies. Okay, thank you very much. And I believe the next subject, because we're leading on from the blood, the very next lecture will be on the immune system.